Our article was titled Population Genomics Reveals that an anthropophilic population of Aedes aegypti mosquitoes in West Africa recently gave rise to American and Asian populations of this major disease vector. Aedes aegypti mosquitoes pose a major threat to human health as they are the most prominent vectors of dengue, chikungunya, yellow fever, and Zika viruses. In 2016 alone, it is estimated that 100 people died from congenital birth defects from Zika. It is important to study Aedes aegypti mosquitoes so that doctors and scientists can adequately estimate the risk and determine prevention methods. Findings from this study contradict the two previous theories about Aedes aegypti. One of these theories is geographically based and states that all the species that are found in Africa are closely related and distinct from the species found outside of Africa. This specific idea theorized that non-African populations were of the subspecies Aegypti aegypti, and all African mosquitoes were of the species Aegypti formosus. The next theory is that domestic Aedes aegypti populations in Africa came from mosquitoes that had migrated back to Africa. This was found because African and non-African and domestic African mosquitoes were found to have different genetic compositions as determined by admixture analysis. Our study argues that genetic differences can be attributed to the change in allelic frequencies that arose from a population bottleneck. This study proposes that the best way to determine genetic diversity is to look for regions of the genome that are strongly differentiated between the subspecies. The purpose of this research is to find out how Aedes aegypti and its subspecies, Formosus and Aegypti, evolved and migrated and determine its historical lineage. The Aedes aegypti populations that were sampled were taken from 5 different locations. These included Uganda, Sri Lanka, and Mexico. These populations are considered to be domesticated because they prefer human blood. 2 samples were taken from Senegal. The Aedes aegypti formosus were taken from the Senegal forest, while the other sample of Aedes aegypti aegypti was taken from the urban Senegal area. Aedes bromelii were sampled from Kenya as a control. DNA was extracted from the individual Aedes aegypti mosquitoes and then sequenced. A pool for each population was compiled. DNA was also extracted from the Aedes bromelii mosquito to serve as an outgroup, which acts as a base onto which to compare the data. Software was then used to compare the recorded pairs between the pools. The researchers determined the similarities between the genomes and allelic frequencies of each population. By doing this, they were able to look at the historical relationships between the populations and more clearly identify the lineage. The researchers divided the mosquito populations into 3 groups based on genome similarities. Group 1 included the Senegal forest and Uganda. Group 2 included the Senegal urban, and Group 3 included Mexico and Sri Lanka. The researchers discovered that the Aedes aegypti formosus from the Senegal forest and Uganda population were genetically similar to one another, and that the Aedes aegypti aegypti from the Senegal urban, Mexico, and Sri Lankan populations were more genetically similar. Previous genetic studies proposed that the African population of Aedes aegypti were genetically distinct from the non-African populations. However, the genome mapping conducted during our experiment shows that Senegal urban is actually more genetically similar to the non-African populations than the African population. Another alternate hypothesis proposed that the anthropophilic mosquitoes all migrated out of Africa, and a few eventually migrated back to become the Senegal urban population. After mapping the genomes of the different mosquitoes, the researchers discovered that the African populations were more genetically diverse than the non-African populations. If the Aedes aegypti aegypti mosquitoes migrated back to Africa, the Senegal urban population should theoretically be the least genetically diverse. Because of the lack of genetic diversity in non-African populations, the authors of our paper believe that the Aedes aegypti aegypti, the domesticated species that prefers human blood, originated in Africa, then migrated to Mexico, and then to Sri Lanka. They believe that the migration out of Africa resulted in genetic drift and lower allele variations. Essentially, this signifies that the urban Senegal, Mexico, and Sri Lanka populations all originated from a common ancestor, Aedes aegypti aegypti, which was domesticated after the divergence from Senegal forest and Uganda populations. This domestication is due to the presence of the odorant receptor 4, an adaptation not present in group 1, which makes this subspecies more attracted to human blood. This phylogenetic tree is a conclusive historical lineage, as determined and proposed by the researchers. At point A, which occurred approximately 10,921 years ago, populations diverged and group 2 and 3's common ancestor is domesticated. At point B, approximately 291 years ago, a portion of group 2 migrates away from urban Senegal and creates group 3. The migration and colonization of this subspecies to different continents increased the global presence of domesticated mosquitoes and subsequently vector-based diseases. In conclusion, we determined that the non-African population of Aedes aegypti evolved from the domesticated common ancestor, Aedes aegypti aegypti. This has a major impact on vector-based diseases due to their attraction to human blood.